Okay, hello, bon dia. Hello everyone, thank you for coming. It is a great pleasure for us at the faculty to have today with us Professor William DeGrado from UCSF. You know that our faculty decided months ago to promote him as a Doctor Honoris Causa from our university, and that will happen tomorrow at the university at the Paranymph. But before that, we were thinking that it would be a good idea that we, as faculty members, can benefit from his visit. Many of you know Bill DeGrado, but for those of them that you don't really know him or didn't know him before these last days, I must say that he is a real pioneer in the protein prediction and protein design field. Actually, it's more than, his lab has been running on more than 40 years. Well, as something as clear as the name of the field, the de novo protein design, is something that he proposed back in the 80s from the last century. And yeah, he had been designing first peptides and then proteins since then. We have learned really a lot about protein folding from his studies. Many of us have followed his work very closely, as you can imagine. And I think it's an inspiring person, because he used to give new points of view for your problem. So it's, if any of you have any problem want to solve with your favorite protein, you can come to him, either in person or by email, I guess. Am I allowed to say that? Okay, okay. Yeah, you can even join, he said. Since we have a long profile to be about him to be explained tomorrow, I'm not gonna get long today, because what I think you want is to listen to him and listen what he wants to share with us. And well, I'm gonna finish now. Just thanking Bill for your generosity to share your time with us. He will be also Monday and Tuesday with us. So I'm seriously saying that if any of you are interested, he will be still around with us on Monday and Tuesday morning. Okay, so. Thank you very much, Bill, for everything, and we are all yours. Thanks.
Well, thank you for the kind introduction. It is a very great honor to be here, and I look forward to meeting everyone. And what I'd like to talk about today is de novo protein design, that is, designing proteins entirely from scratch rather than by modifying natural protein sequences. I remember my last year in college, I learned two things in my classes. One, I saw the first structure of an enzyme. It was lysozyme had just been solved. And I thought, wow, that's great. I'd love to be able to design something like that. And then shortly thereafter, I learned about Merrifield's solid phase synthesis. I thought, wow, I could actually make one. And so now it's taken over 40 years, but I'll tell you today, hopefully we'll have enough time to talk about designing enzymes. So our outline is, first we're going to talk about designing proteins from scratch using physical principles and self-assembly. So things that look like this. That's sort of more historical. Then we're going to design proteins to test principles of small molecule binding and how one protein scaffold can recognize many small molecules shown there, and then finally designing enzymes. When we started out, the big everybody thought we were crazy. They argued that this could never happen, and the types of arguments they gave was the sequence problem. If you think about all the numbers of sequences for a 100 residue protein, that would be 20 to 100. How would you enumerate them all and find out which one was right? Conformational problem was also a big issue because for a protein of 100 residues, you've got 10 to the 100th possibilities. And then temporal, how did it ever happen that it could fold on the time scale of the universe? And yet, when I thought about it, we have 26 letters in our alphabet. Does that mean we really need to enumerate every combination of characters before we could derive the meaning of a sentence? I don't think so. And so proteins had evolved, and so it seemed it couldn't be that hard. And so the first thing people were thinking about that time was that maybe proteins evolved from short random peptides, and then these sequences could self-assemble into helices or sheets, and then they could, through gene duplication, become one protein and asymmetrically optimized for different tasks. And so with that in mind, we started our work, and the first thing we thought about was how do you stabilize secondary structure? And so we found if you put hydrophobic residues that match the periodicity of a helical structure or a beta sheet structure, then when you allow them to self-associate, they perhaps could make something like that, a helical bundle. Or if they were more like a beta sheet, then we could make something like this. And so this was news at the time. And we thought that presumably, because a lot of thermophilic proteins, the most ancient proteins, do in fact have structures that look very repetitive, a lot like that beta sheet structure that I showed you, that perhaps those were the first enzymes as life arose. And to test that hypothesis, we made a very simple heptapeptide shown here with two histidines that can bind a zinc, and when they bind zinc, they catalyzed hydrolysis of CO2. And they also self-assembled into rather large structures. Using that type of approach, we also thought about how could it be that more complex structures that don't have translational symmetry, but instead have closed symmetry, might have evolved. And so here is a deconstruction of rubridoxin, an iron-binding protein, and of diiron proteins shown here. We start with a helix, and then that brings us to here, and then ultimately you evolve into the full enzyme. And so if you think about that, then you could start with, here are all these diiron proteins, very different structures, but embedded in them is this very simple 4-helix bundle, which then is even simpler because each one of these helices has a glutamic acid as a ligand. And so we can basically have a mathematical embedding that takes us from here all the way up to here at least, and then beyond. Sounds very familiar to modern work in AI. So we start with something very simple. We apply translations, rotations. We end up with something like this. And then it worked very beautifully with my colleague, Angela Lombardi. We had a number of papers about these. And we were able then to evolve from that very simple structure to proteins that catalyze 2 electron oxidations of oxygen or 4 electron oxidase reactions. But again, this is a while back. And finally, we were able to design porphyrin or heme-binding proteins that were adjacent to the dimetal binding proteins. And what we found was that as we bound metals up here, it transmitted the information to the catalytic site, and so that we were able to see allostery between these two sites. Here's the oxidation rate in presence and absence of the porphyrin. We're also able to design rubidoxin-like structures. Often people ask, have we ever designed anything but a helical bundle? I think I've shown you a few examples now. So now we jump forward to the modern era, a month or two ago, and I got interested in this problem again. And at this stage, what we can do is we can enumerate every possible repeating pentapeptide that is possible. Just enumerate them all. There's less than 1 million of them. And then we can predict what their structures are using things like Raptor X, Boltz-1, or AlphaFold 3. Usually we do all three to see if we get the same answer. And then we discover which of these scaffolds were very, very highly designable. And what we see coming out then is, so here's a histogram. We're looking at the propensity of something to form a folded structure. And this shows the fraction that can fold to a very high, this is the frequency, and then this is the probability of their forming a folded structure. Anything to the right of this line here is likely to be folded, very, very likely. And so what we find with a repeating pentapeptide, we get about 2 or 3% are able to fold into a structure that we'll see in a minute. 6-mer, maybe a little more. It reaches a maximum around 10 and then starts to fall off. And indeed, by the time you get to be a large protein, even 30 residues, then the probability of folding becomes extremely low. So this really validated the ideas about simple peptides being repeated, and it's easy to think through every possible pentapeptide. And so now if they're repeated, you can get something that's folding, and as we saw, began to function. So what do they look like? You can see here sequence logos. It's work that I love doing on the weekend, so sorry to take you through this at such gory detail. But this is what you can get out of a 5-mer. 2 structures look very much the same. This shows the probability. They all more or less look the same. You see sequence logos, and this logo matches very precisely the logo that you see in modern day 5-mer repeat proteins from archaea. And we get a different one here, and we can see using a Ramachandran analysis precisely what makes them work. Going to a 6-mer, now we find that it makes a more tightly wound triangle in this case. Again, very strong sequence logos saying precisely what it is that makes it work. But now we can see that there's other clusters that are possible, different structures. And then by the time you get to a 12-mer, we have many different structures, all possible. And with different sequences, we can get different sequence logos. And at this point, a fair amount of structural diversity. So this shows the top cluster, and then here's a second, here's a third. So by searching through these repeat proteins, we get increasing complexity as the repeats get larger, and we hope in the very near future to start making these and showing how they can be functional, both as materials and as catalysts. Another way that we're working with some of these repeat proteins is to make biosensors for phosphorylation with Xiaokun Xu at UCSF. And the idea is we start with a protein, small protein, that is fused to a fluorescent protein. And then in response to phosphorylation, we can cause them to self-assemble. The details really aren't that important. But what's important is that when you phosphorylate, now we make a site for binding that phosphoserine, and then it becomes punctate under the microscope. So you can see it as you turn on a kinase. Now what I really like about this, unfortunately the movie didn't show, but here's a time-lapse sequence. After we turn the cells on, we're turning on protein kinase A, and here's the receptors, and you can see that the signaling, we're looking here, see this shows signaling growing, 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 growing. So not only are we able to see that we have turned on pKa, but we can see precisely where we turned it on, and it's already turning out to be a useful tool for other people who are interested in organelle signaling. So we also can make ones that respond very quickly. When you add an antagonist, they go back to baseline, and others that are more integrative. So they basically stay on and you can look at them later. And so we've done this for ERK and PKC as well. Okay, so let's look at how proteins bind to small molecules. This was a great problem in terms of it's easy to see them. It's easy to break proteins that bind small molecules, but can you design them from scratch? That turned out to be a much harder problem than just designing proteins that folded. But beyond that, of course, you have to get funded to do this, so why might you want to do this? Well, you can imagine them being very good antidotes or reversal agents. So if you have an overdose of a given drug, now you can bind to it and reverse it, and we'll see that. Secondly, we can use it to carry drugs with very poor pharmacokinetic properties. Here in a school of pharmacy, this is something that really makes all the difference between a drug and a not drug. Imagine also putting in the loop now would be a receptor binding domain. This is, in fact, somatostatin binding to a somatostatin receptor that's very rich on cancer cells. And this could then be an anti-cancer drug. When it gets endocytosed, we encounter cathepsins that clip it, and out comes the drug. So these are the types of things that we want to be doing with these molecules as we make them. And so how does it work? Well, in the modern era, we use AI methods basically to predict protein structures and design protein structures. Diffusion algorithms are used mostly. And in these, we start with a three-dimensional structure of a known protein. And then we start adding Gaussian noise until we have a completely random structure here. Okay? And now you memorize the gradient, the direction that you have to go to get from here to here, and then you go in the opposite direction. You've done this not just on one protein, on many, many different proteins. So basically, you can start anywhere with a random structure and say, oh, make me something. And one time it might make this, another time make a helix bundle. It's all very stochastic, but all the instructions are there. And most importantly, the gradients, that is the direction you need to go to get something that's very native-like, is also embedded in these models. Now, so this is what it looks like. You start with something random, and then boom, with time. And this is taking maybe minutes on a computer nowadays. And from Gregorian's lab, they showed that you could design proteins with constraints. Here's the alphabet and numbers, and they actually have shown that this works. But when it comes to designing small molecule interactions, this was a lot more difficult. You build graphs for not only the protein, but the ligand, and then the ligand graph has to talk to the protein graph. And while it works to some extent, this is a work from David Baker's lab, and this is really typical of what they see. So if they want to design a protein that binds a small molecule, they'll make on the order of about 10,000 proteins. That's a very expensive to buy all those genes. And then they express them in yeast, and then they look at the ones that bind. They may get a handful. And then they have to optimize those, so they redesign them again, another 10,000 genes. And then ultimately they get a few binders between low micromolar to mid nanomolar. And our approach is, well, why is it so hard? Why aren't they doing better? And if you look at a real protein, you really get clues. For one, there's this cooperativity problem. All of these interactions occur simultaneously, and you might have repulsive interactions between these residues that are binding to the small molecule here. It's hard to think about how do you position something that's destabilized so that it can be stabilized once the small molecule binds. So it's a sampling problem, and it's also a scoring problem. To alleviate this problem, Nick Polizzi in our lab developed an approach in which he made what we call Vandemers. So a rotamer is if you specify the backbone position in a protein, then it says, if I have a given rotamer, then I know exactly where to position the side chain atoms. And so we thought, why don't we do this in terms of, here's the backbone, here's an amino acid type. Now, where does the interacting group locate? And so what this allows us now to do is, given a backbone, we can search for sequence, but also very good interactions to bind small molecules. And so we did this. The paper's published, but we have looked at anti-cancer drugs. These are PAR-P inhibitors with Alan Ashcroft's lab. And we tried to bind to this common pharmacophore in all of them, but not, say, in these. And indeed, we were able to design proteins that engaged all the polar groups and bound with very low nanomolar to subnanomolar affinity. And they also worked in vivo. In this case, they're neutralizing the drug. Oh, and one thing that I'm most excited about is that although this is all using sort of AI-type approaches to design, we can also use physics-based approaches to predict what the binding energy is. So this is a number of different drugs and a number of different mutants, and we're predicting using physics-based methods, MD, what the free energy of binding should be, and we get it to within a kcal per mole in absolute free energy of binding. So that's quite extraordinary. Do they work? Well, we can take our proteins. Everybody worries, and for good reason, that the protein might be immunogenic, so we make it in all D form. And that way it can't be cleaved and then presented via the MHC to the T cell and get an immune response. And we also make cyclic lengths. And then we put them into animals. And we can see first, this is apixaban injected into the animal. And then this is what we see if we have apixaban plus our small protein. What happens is it accelerates the clearance, and it does so by just being filtered as a complex in the kidney and then excreted into urine. Here we can look at the immune response, and so if we have an all-L protein, this, by the way, is a apixaban binding protein, which we call ABL, and we can see here's our binding curve, or here's our immune response. On the other hand, with the all-D form, we could not force an immune response. So we're able to design proteins that bind small molecules. Let's come back and think about evolution and how you evolve specificity from one protein or one small molecule to another. And Dan Tawfik spoke about how you have specialists that can bind to one molecule quite tightly. But it might also bind or catalyze another reaction very weakly, but specifically. And so this is what you call sort of moonlighting as a different function. And so you've got not only your specific interactions going on and catalytic processes, but at a very low background, you have all these other things that they're able to do. And now so they've come way down the fitness landscape. But now if you have a selection that will say, oh, I really need to bind B, then the protein can mutate, change its shape, and now we have a new fit protein that can bind to the new molecule there. So can we do this? And this is work that's done by Yuta in our lab. And it's in collaboration with Jamie Fraser's lab at UCSF with Lena Bergman and Galen Corey. And I know all these names don't mean anything to you, but they're very important to me because they do the work. Anyway, so when we do de novo design, we look at a very immense sequence space, as you saw, millions. And yet we usually only look at one or two molecules. And so what he wanted to do was say, if I have that protein able, can we not only look at sequence space, but can we look at chemical space and find everything that it can bind within a reasonable approximation? And then once we find something, can we make a new specialist? And then if we have that specialist, how much or how special is it? Is it really specific? And so we go back and forth between exploring sequence space, chemical space, sequence space, chemical space. So let's start with the sequence space. This is our able protein, and it binds to apixaban. By the way, we chose this molecule just because a cardiologist on our floor said he wanted a protein that would neutralize this blood thinner so he could get patients into surgery more quickly. And so here it is. It encapsulates that protein. And what we did next was to challenge that same protein now with about 500 small molecules and determine the structure of anything that makes a complex. You do this by crystallizing the protein, and now we can diffuse in one at a time these molecules, and then collect a new diffraction pattern and see if it's bound, and if it's bound, then we get the structure. Okay, so we see what sticks, and we get 43 hits. So this is a design protein. That number is about the same as if you were to work on, say, the macro domain and see how many you get. So hit rate is very similar. That means that they're very similar in specificity. But when we look at what happened, how is it that they bound these fragments of small molecules? This is what we expected to see. So here's a tyrosine interacting with the flat surface of a ring. And looking at it, here's this ring in an aromatic box. But if that tyrosine just moves over a little bit here, and this isn't even a different rotamer, now you can get the small molecule on the other side of the tyrosine in a completely novel interaction that we had not anticipated. And so now we've found that it binds this molecule, which looks a lot like turn-on fluorophores. So we said, could we make it bind to this molecule? And to do that, well, here's our apixaban here. This is the fragment that we found here that binds weakly, but specifically in terms of geometry. And then the molecule we want to bind is larger, and so we put it here in the same spot, but we redesigned the protein to fill up that empty space that was occupied. And having done this, we find that indeed it binds. You can see a fluorescence increase. Here's our binding isotherm. We then can go back to chemical space and we say, let's look at many, many variants of this fluorescent molecule. We go to my colleague Jason Gestwicki at UCSF. We collaborate a lot there. And we look at, do any of these other ones, molecules, bind to our protein? And there's far more than what is shown here. And what we find is only two, and they have the same framework, more or less the same interactions. So indeed, we've gone from a specialist to a generalist to, again, a new specialist that's quite specific. Okay. So let's now think about, given that ability to bind fluorophores, what could we do that's useful? And we sat down with Bo Huang, professor at UCSF, And Dyke Mullins, again, another professor, and said, well, what is it that we'd want? Well, because we'd like to make molecules that would be just like GFP, but now we can add the turn-on fluorophore, and we can use them in the same way that use fluorescent tags. And so this will give us a degree of orthogonality compared to everything we can do already. And so it should be very efficient in its labeling. We'd like our protein now to be much smaller than GFP or SNAP tag or any of the other tags, so they're less perturbing. What most important, though, was stability to temperature. I didn't realize this at the outset, but thermophiles live at 75 degrees, and so all of our existing tags like GFP and SNAP tag and halo tag don't work. So you can imagine trying to do all of biology in thermophiles without fluorescent tags. It's unthinkable. And I had no idea that existed. Okay, it has to be orthogonal to other methods. It should be very fast. As soon as it binds, it should happen. And then it should be very, very bright. So for the very bright part, we worked with Luke Levis, who makes near-IR dyes that are very, very bright. And so these now became our targets. And that's a lot more complex than what we were dealing with before. Nevertheless, by more or less the same process, Yuta was able to design these proteins that bind in the nanomolar range to the molecule. By the way, for single molecule work, this is technical, but you don't want them to bind too tightly because the ideal is for them to come on for a while, come off, come on for a while, come off, and that allows you stochastically to get super resolution. So anyway, the affinity is about where it wants. And all of these proteins don't unfold all the way up to 95 degrees C. So out of 9 proteins that Yuta designed, 8 worked. And when I say worked, this is now trying them out. This is bound to actin, or fused with actin. And you can see really stunning images. This is STED. And here's where you can see single molecule imaging. In this case, it's bound to a histone, or fused to a histone, excuse me. And you can see walking through the beautiful images that you're getting of single histones. This really makes our coworkers really excited. And so they put it through the paces for thermophiles, and they've put it for nuclear localization, nucleolus, lamina, actin, microtubules, etc. Each case they were able to succeed with multiple proteins, and you see them very nicely live as well as fixed. Finally, we were able to show, I won't go through this, but basically what it shows is that we get brighter labeling than one can get even at room temperature out of halo tag, and that it's quite orthogonal to halo tag, so it can be used together. So this again shows a confocal image of one of our proteins, and now look at here is STED. Come on. Oh, the movie doesn't show. Anyway, you can see how much sharper. This is much sharper than that. It's a little hard to see from the image. There's a different. Okay. But how do we do in terms of temperature? Remember, we're trying to work on these thermophiles that live in deep vents like this that we have in California, and you can go visit. And so these are 75 degrees. And to do that, Dyke Mullins' lab attached our protein to a membrane protein or to a DNA binding protein shown here. And now we can look at images, and you can see this is nuclear imaging or DNA imaging, and then this is the membrane. And so this is the first fluorescent image, a thermophile, as it reproduces, as it grows. Come on, let's go. There we go. See, it looks kind of similar to everything else you've seen, but the collaborator was just so excited because he'd never seen a fluorescent image for a thermophile. And so, this is the group. I've kind of called people out as we go. So let's get on to designing enzymes. And can we do not only enzymes that catalyze natural reactions, but we're going to be asking, can we catalyze reactions that aren't seen in nature, where we're making multiple chiral centers. And this work is done by Sagar and Yuta. And we started with a very simple benchmark reaction that people have been working on for 40 years, starting with catalytic antibodies and then design proteins and so forth. So people like Pete Schultz, Don Hilbert, David Baker, everyone's worked on this reaction to see how good their catalysts work. And so here it is. And we're taking a CH bond and we're removing a proton. So that's very much like triose phosphate isomerase. We have a transition state analog that we can design against. And in our protein, we had a histidine that interacted with carboxylates when we looked at everything that the protein could interact with. But we changed the histidine then to aspartic acid, and then it was just in place to bind to the transition state analog. And what we found right from that design, comparing to the computer designs of others, including Cyril Fleischmann's this year, we find that we're 1 to 2 orders of magnitude higher. Now, the next step is always directed evolution. One changes all the residues around the fluorophore in site saturation mutagenesis, or fluorophore, the small molecule, and then sees which ones get better, and then they combine them. Francis Arnold got the Nobel Prize for that work. And so after one round of this, we're getting up here, which is about the same as triose phosphate isomerase. This shows our others that have worked on this and how far they went on a logarithmic scale. Next, we look at the NMR spectrum of the protein. So every dot here is associated with an amide NH in the protein. And what you want to see is that it's well dispersed, lots of dots. And it is, but when we add the transition state analog, we see movement of these peaks. And some of them aren't right around the small molecule, but instead they're more distant. And so presumably these are allosterically important. They help position the protein just right. And so when we mutate those, now we get all the way up to here. So it's now getting very, very active. And this is, for example, where we are, and this is Hilbert's protein after 17 rounds of directed evolution. So what do we learn? We were all about rational stuff, right? And being able to predict things. And now we go and do some irrational mutagenesis. And what types of things do we learn from that? Well, we learn things that we would have never guessed or never tried if we were smart. For example, this was a leucine here, so middle-sized residue, and it turned into a big tryptophan. Our algorithms would say, oh, the protein's not going to take that. It's going to have a really hard time. AlphaFold tells you, oh, it's really now distorted. And yet the protein is able to make small adjustments and allow it to happen. More interestingly, here, right in the middle of a helix, next to the substrate, we have a proline. So anyone who's taken biochemistry probably knows that prolines are helix breakers. In our computer algorithms, we never change a residue to a proline within a helix. And yet that turned out to be what made it better. And here's a water molecule that inserted into the exposed amide that was created. And that seems to be very important for catalysis. Finally, we can ask, what is it that's happening? And these show torsional angles of residues in the protein before and after our optimization. And this shows the number of water molecules. So it is greatly dehydrated. It becomes dry in the active site in the highly evolved proteins. So that was kind of a warm-up with a benchmark reaction. And now we wanted to say, what would be the ultimate goal? And if we start to think about enzymes for a greener world, we all love our synthetic stuff, right? The carpet, the thing you're sitting on, etc. My shoes. But these are all done in ways that are suboptimal for the planet. Could we do these by enzymes in water or at least very green solvents? So that's a part of our lab is starting to think about that. Now, to do this, we aren't limited to nature's cofactors, so we can make artificial cofactors at will. We want to keep the protein small because that means it's atom efficient. It doesn't take many, many grams to make a small amount of catalyst. And in terms of working, we're used to our enzymes working at micromolar substrate concentration because that's the concentration they are in cells, right? And so they have to have deep invaginations and binding sites. And yet when we're trying to do a chemical process, we have our protein our substrate at about 1 molar. About 10% to 20% of the solvent is actually your substrate. And so that is sort of a real changer in your mindset of how are you going to design these things. People will always look at this and say, oh, it doesn't have a deep pocket. Well, it doesn't need one. They can even be detrimental. We'd like them to be evolvable. You just saw the power of evolution. So we'd like to design them or evolve them. We also saw that we could make things very thermostable for reactions if we have to heat them. And then another very important thing is stability to organic solvents. I think you know that if you drank enough to reach 50% ethanol, you would be quite dead because your proteins would not be happy with this, but we have to design proteins that can live in solvents that high. So the reaction we're looking at is one that was popularized by Francis Arnold, and we take this styrene and we add a carbene precursor, and then we make 2 single bonds, and it creates 2 chiral centers. So the game here is, can we make only one of these 4 isomers? And this is done with a good friend and collaborator, Yang at UCSF, and Peng Liu, who's a quantum theorist at University of Pittsburgh. And so the background for this reaction is it has been done by, as I mentioned, Francis Arnold. David Baker also showed this, that he could do this, but his EE, enantiomeric excess, was 93% and about 5,000. So this small molecule models can do that. And what they noted in their paper was that de novo design efforts had focused on helical bundles that would never be able to catalyze reactions. So I thought, that's interesting. And so we went and took one of our porphyrin binding proteins shown here, and we wanted to make a binding site for the substrates. Now, it took a lot of convincing to my postdoc to cut away the active site so that we would have this opening, because we're all thinking again about deep binding pockets. But this was just the deal. We designed it, and it showed, well, now we're doing okay. on the diastereomeric ratio of those molecules. But when we solve the structure or predict the structure by AlphaFold 3, we see something like this. But when we actually solve the crystallographic structure, we see something like that. So we say, okay, no wonder it's not superb. It's just okay. That's because of what I like to call the final angstrom problem in de novo protein design. So if you were to look at a paper, we were to publish a paper on just designing a protein, and it came this close to our design here, we would say that's fantastic, it's within an angstrom, it's done. But for catalysis, you really need to get things with sub-angstrom accuracy, and you need to get it right. So that means you have to pay attention again to the physics, and in this case, using QM, quantum mechanical methods. Okay, so we went back to really old school methods that Gregorian had developed when he was in the lab to find loops that were better accommodated in here and extending the helix, and now it works beautifully. 4 out of 10 of our designs gave more than 99% of the proper isomer and just one. And we were able to make all possible isomers. Okay. Now, what's the point? So in summary, we made things that also have 40,000 turnovers. That's a very big number for synthetic biologists. It matches the most successfully engineered proteins, and it's stable up to 70% ethanol. So if you're going to use an organic solvent to dissolve things like this, ethanol is your solvent because it's the greenest of all solvents, and it doesn't lose any activity. Finally, we can look at other reactions not catalyzed at all in nature. Here is a silyl insertion reaction. Our protein just off the shelf that we had designed, by now we had a large, small library of them, so we can look at them for other reactivities. So it was already showing modest activity, but after a little bit of design, we got it better. But then after a large campaign of directed evolution, we got ourselves to a protein with 99% of the right isomer, and it's able to get 32,000 turnovers, and it's stable in methanol in that case. And so what did we learn again from the random mutagenesis was, again, we found a proline right in the helix. So the types of things that are hard to anticipate, and again, are helping us solve that sub-angstrom problem in design. This is probably more than we want to see at this hour. So in conclusion, I think when we look at designing proteins or taking the results of AI, a mixed initiative always works best. Basically, this gives us the tools to enhance our own understanding. And it's not going to replace it, but instead give us these wonderful tools. We always find that a mixed initiative works far better than black box approaches, which means we still need biochemists and we still need chemists because computer scientists can't have that intuition of how these things should really work. And then when it doesn't work, of course, if you've got a biochemist, they can say, well, that's because of this and this, and we need to bake that human intelligence into the artificial intelligence. But we do need to always stay one step ahead of AI, and I think protein design will be increasingly important and practical for environmental applications. We've also seen healthcare applications I didn't mention. And here's all the people that have worked in my lab over 40 years. So thank you very much.
So thank you very much, Bill. You know, you probably realize it's a good time to design proteins, so we're still all in the good place. Does any question?
Yeah.
The
Didn't
audience?
you say you were going to buy dinner for any student that asked the question?
Okay, okay, we can do that. If any student asks a question, we invite him for dinner.
They think it's McDonald's.
Maybe I can start, maybe.
Okay.
Yeah, I see that, you know, this targeting approaches to release some small molecules to specific cells. It's really a funny thing. And the other great, you know, focus is on designing new enzymes. The question, just to be more closer to your nearest work, are you going to focus more in the novel design of enzymes for, you know, environmental purposes, or more for the health approach, you know, to use protein design to target small molecules to specific cells to treat a specific
Of course, a lot of this depends on my collaborations with organic chemists and their needs. With Yang, we're just trying to do basic reactions much more efficiently that medicinal chemists would do anyway, day to day, or for processes. We're also interested in photodecarboxylation of fatty acids, which you can get from soybeans, into hydrocarbons, so it relieves part of the requirement for petroleum. It still is making carbon, so it's so-so. We also are hydroxylating these substrates for lubricants, and so society has many different needs. Maybe the most interesting is this one with a company, and they want to make channels that allow aspartic acid to come out of cells. So it turns out when you make small molecules in synthetic biology, you can really turn on metabolism, but then you make so much of the product that it's toxic to the cell. And the rate limiting step is just getting it out of the cell. So if we can make selective channels for this, then we can make aspartic acid. Why do we want aspartic acid? Well, polyaspartic acid can replace polyacrylates. Polyacrylates are used in paper diapers. And so, you know, they absorb huge amounts of water, but they don't break down. So those diapers are all sitting somewhere in a dump, whereas polyaspartic acid actually does break down. So I think it'll allow us to make biocompatible substitutes for polymers.
That's clever. Any other question?
Hi. You mentioned that you try to keep your proteins, your designs small, as a small protein, and apparently you can do many things with these small proteins. So why do you think, or what could be the advantage of what we see in nature, that proteins are probably, in average, much larger than your designs?
Yeah. So there's the evolutionary reason for that is that by having a large target, you can have mutations all over the place, and the protein more or less can stay intact. And so you can have all these neutral mutations without anything bad happening. Whereas when it's a really small target protein and you make a mutation, then it can have a large effect. So that's one reason. Second, you can imagine if I was trying to make a specific ribonuclease here, that it would be nowhere close enough to be specific. Maybe it would cleave RNA, but it would not be specific. So substrates do get larger and larger And so as we think about substrates that are larger, then we have to think about larger proteins, which we're doing.
In the case of the Kemp eliminase reaction, your design, so people have been using concepts such as the substrate repositioning or electrostatic stabilization of the transition state to explain improvements. So why do you guess that your de novo design is so successful?
Yeah, gosh, going all the way back to Perutz, you talked about the importance of placing the substrates in reactions like this in in solvent inaccessible regions. So not just binding the transition state, but binding it in the right spot. And so we put it in the bottom of a deep tunnel. The first one we made, actually that aspartate pKa went all the way to 10, so we kind of had overdone it a little bit, and we had to back off. But I'm rather certain that was the part that was missing. This is what Warshall and Head Gordon had argued rather vorifus stringently. And so it had been an argument in the field. And so, but it was amazing when we just used common sense, things worked really well.
Hello.
Hello.
That was very cool. My question is about the weird cases that you found where, you could have, for example, prolines that you would normally expect to destabilize a helix or something that were important for your function. Do you think you're seeing these because you were kind of outside of natural sequence space and you can find sort of more alternative solutions than you would in a natural protein? Or do you think you could also have this in nature?
Yeah. In part, yes, our proteins are so hyperstable that you can take a mutation that would, whack it by 3 kcals per mole, a substantial destabilization, and still can stand it. It can still be folded. Also, in natural proteins, you'll find in helices extremely conserved regions where you always have a proline, maybe a serine substitute, but always a proline, especially in transmembrane proteins. It's well known. So, yes, sometimes you have to pay with stability for function. Excellent questions.
So, somehow related, Bill. What about using artificial amino acids? I mean, non-natural ones, in the de novo design? Can we use the skills of AI to try to figure out if non-natural amino acids could improve some reactions? Or
yes, yet another great question that you set me up on. And so, indeed, we're working precisely on that. Hope to have our first data very soon.
I didn't know. It was not. Okay.
Okay, you're going to have to buy lunch.
I was curious, in this approach, you go from a specialist to a generalist binding. You try to find all these, what finds different, and then change the protein. So it's a specialist for that kind of molecule again. In the process of choosing what is going to bind, like what's the plasticity of the generalist approach? I mean, what kind of molecules you try? You try similar molecules with different groups. At what point you change the first molecule? Do you get my point?
Yeah, okay. Yeah, absolutely. Again, excellent question. We use a diverse set and then see what will stick to a given site. What we're doing now is designing not just this one helical bundle, but a lot of different topologies, and then we'll see what sticks into them. We're also enriching the library, this is with Jamie Frazier, with transition state analogs, so that we'll already have our foot in the door and make it a little easier for design.
Thanks.
Thank you.
Thank you.
Hi, thank you for your great talk. I had a question regarding the design of non-immunogenic proteins. What about, you mentioned at some point something that you did in order not to, can you repeat that part, please? I didn't get, that you designed the proteins that you could administer to mouse, or I don't remember at this point. Were you actually sequencing the sequence, so were you actually like sampling the sequence space that could a priori have no binding with MHC, or you actually did some kind of a biological transformation after you designed?
Thanks. What we did was to inverted the chirality of the protein. So it's all D amino acids. So proteases will not work on it. Even if you get peptides, they bind stereoselectively to the MHC. So that works with a drug like apixaban that has no chiral center. If it has a chiral center, then we have to actually consider chirality when we make the all-D version. So you chemically synthesize this. Actually, Bobo Wang did this at Westlake University.
Do you think it is a good way to think about the design of proteins that couldn't cause no immunogenetic response, to like only selectively sample the sequence space, or somehow to manage to couple it with MHC binding predictions?
Yeah, the de novo protein that's gone furthest in the clinic is used for T-cell therapy. It came out of this little 3-helix bundle that we designed 20 years ago, not knowing that it would be useful for something. And the company that did this, Arcellx, spent about a decade scrubbing that protein of all MHC epitopes until they got some completely non-immunogenic. Now it's much easier to predict immunogenic sites and MHC binding sites, but it's still an effort unless you can chemically synthesize them with reverse chirality.
Okay, thank you very much for your talk. My question is, what is your opinion about this so-called mirror life that was recently in Science? Was a paper talking about the possibility to build artificial cells entirely with the stereochemical, opposite stereochemical versions of every monomer. So would you think that in our world, in our biosphere, would be possible to have this kind of mirror life?
I don't think so. It is so hard to do what these people do because chemically they have to synthesize every single molecule to just like get together a protein that will replicate DNA. And so that takes years of effort. And then they also have to, the DNA has to be synthesized. And so we might see things that could replicate in a test tube, but going beyond that to life is a pretty huge hurdle. I'm a lot more worried about many other things, like Trump.
Thank you.
So thank you. Any other further question? Suggestion? No? If not? Okay. So then thank you very much, Bill, for such an inspiring talk. And to all of you, thanks for coming. And those of you that will be at the Paraninf tomorrow, we will join him again. So thank you.